Welcome to muscle physiology, part 2 or sort of, and I'm going to try to explain the sliding filament theory, and hopefully I don't mess myself up too much. I'm a little rusty, and you think, well, maybe I should have studied more ahead of time. Yeah, I should have. I was having technical troubles that I won't even go into, but I'm finally recording, I think, and the audio's okay and the video's okay. So here is just a look under a light microscope of skeletal muscles, cells. As I mentioned, one cell runs the whole length of the muscle. So whatever muscle you have, well, so the longest muscle cells in your body will be these in your legs, maybe the sartorius muscle, because it stretches across. So you'll have some really long cells, but the cells are multinucleated. So they're one cell because they're all contained within the cell membrane, but it's kind of cheating because you have lots of nuclei. And the nuclei have been kind of pushed out because the muscle's just very specialized and just full of these contracting fibers. Now, muscle cells are similar to other cells, but they have weird names. Just because. So we'll see that. So here's the whole skeletal muscle, and it's got many bundles, and a single cell or fiber, muscle cell, are the same thing and consists of many myofibrils. And when your muscles get bigger, you don't add cells, but you do add myofibrils to cells. So muscle cells are packed with lots of mitochondria. They have a cell membrane, but instead of calling it the plasma membrane, it's going to be referred to, boy, I'm tired, aren't I? Blah, 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 blah. Instead of the cell membrane, it's the sarcolemma. Sarco is referring to flesh, sarcolemma. So instead of the plasma membrane, it's the sarcoplasma inside. Instead of plasma, sarcoplasma. Instead of the endoplasmic reticulum, the sarcoplasmic reticulum, and so forth. So the endoplasmic reticulum of muscle cells, the sarcoplasmic reticulum, it's quite a bit different than a regular cell. It's very extensive, and it has these transverse tubules as part of it, and they're going to be important in spreading the signal from the nerve cell, which we'll see in a little bit. So the components are kind of the same, but also kind of different, so it's a bit confusing. All right, this is more of the same, but the muscle cell or muscle fiber with all of its myofibrils, and then looking more closely, the pattern that we see we have different bands. We have kind of wide bands that are dark, and then lighter bands with a thin dark band in the middle, and we can kind of see that here. Well, we have wide bands with dark. Now on this, all we just see these dark lines, and with space in between. Those are our Z lines. And so from Z line to Z line is the length of a sarcomere. So back to here, this is a sarcomere from here to here, and that is what is contracting. Just the two Z lines are coming closer together, and you do that along the whole length of the muscle cell, and the whole muscle cell just gets a little bit shorter. That's all it does. So we're going to be looking at how does that happen. So a sarcomere is the space, one band, one unit of muscle cell. And what it is, it consists of an array of thick and thin filaments. And of course we have more words. The thick filament is called myosin. and it has these little globular heads on it. And the thin filaments are called actin. So we have actin and myosin filaments, and they are going to interact, but not all the time. And what's going to determine whether or not they are interacting, whether the myosin's grabbing onto the actin or not, is this little rope-like thing, protein, on top of the actin filaments. And that's called tropomyosin. And whether it is covering the binding sites or leaving the binding sites uncovered is based on whether there's a signal from calcium binding to what's called troponin. So we'll go through this step by step, but basically calcium is going to get released from the sarcoplasmic reticulum in response from a nerve signal will cause that. The calcium leaks out, or floods out rather, binds to troponin, slides the tropomyosin out of the way, which exposes the binding sites. The heads from the myosin automatically grab onto those binding sites and start to ratchet. And so the heads on this side will pull this direction, inwardly this way, and the heads on this side will pull inwardly this way. That's it in a nutshell. We'll go through it several times. Just another look, and we can learn A band and I band as well. I'm just not going to go there. And an H zone. I'm just going to say Z line to Z line is a sarcomere. And what we see, there's this area right in the middle where there are no globular heads, and then this darker region here, that's where we have actin and myosin overlapping each other, and then we have this region right here where it's just actin, not overlapping with myosin. We have the same thing on the other side, and then we have a little connection, a Z line. Now, as the muscle contracts, I mentioned the Z lines come closer together, and so this band here, where it's just actin, that's going to get narrower because the actin is going to be pulled along the myosin. It's going to slide along there. So this little region and this little region are going to get narrower for every single sarcomere. Or every single, sarcomere, that's the right word. Little unit of contraction. So, striated muscles, and I could be wrong in saying that those dark lines are the Z bands, but because it could be lying, but I'm going to stick to that story. Either way, sarcomere, 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 lots of repeating units that get closer together. So there again, there's the sarcomere. Here's the region with just actin. Here's a region of overlap. Here's a region with just myosin. That overlap region will get bigger as the non-overlap region gets smaller. And the software, the physiology software, has this in video form, and you'll want to check that out as well. And so every single muscle fiber, which is one cell, very long cell, gets a stimulus from the ending of a nerve cell, of a motor neuron. And the motor neuron will have a place along the plasma membrane, the sarcolemma, where it releases neurotransmitter. And in response to that neurotransmitter, the signal will get carried into the muscle cell, and the whole muscle cell, all these little myofibrils that make it up, see, it's many myofibrils together, and then we're seeing that banding again. But it contracts, the whole thing contracts fully, and it's just a quick twitch unless it keeps getting more nerve signal. As long as it's getting a signal to contract, it will still, keep contract, and it can stay in a state of contraction. But otherwise, it just contracts very quickly and relaxes very quickly. 40 thousandths of a second or something. It's very quick, but it gives it its full contraction or none at all. So if you want more strength, you just have to engage more muscle cells, because any one muscle cell doesn't give just a half-hearted effort. It goes full contraction for the time it's getting the stimulus, or none at all. So here's that actin filament, and it's got the tropomyosin on it. It's associated, and these are just repeating, kind of self-assembling proteins themselves. Here's the myosin with the little globular heads, which can pull, which can ratchet. Here's a little connection from one, between the Z lines is the sarcomere, and here's where the middle of the myosin, and the myosin is this darker, thicker filament. This red region, which is probably what we're seeing under the microscope, I confess, that's probably what we're seeing. That's the region of overlap between actin and myosin. And we're drawing in 2 dimensions, or we're showing 2-dimensional pictures here. Keep in mind that one myosin is surrounded by like 6 actin filaments, not just 2. So it has one here, but also one here, and one on the other side, and a couple of others. So I don't think we need to know the names of all the bands to understand the overall picture. Okay? And I think this makes it clear enough that Z lines are brought closer together as the actin filaments get brought towards the center of this whole thing. So even though the muscle cell shortens, none of the filaments get any shorter. Right? None of the filaments get shorter. They just overlap each other more, which is an important point, I think. So here is at rest, and here is contracted. At rest, contracted. So what changed? Well, the shape of the Z line changed, but more importantly, the amount of overlap changed. So this is very subtle here, but the actin filaments were pulled closer to the middle of each sarcomere. And yes, some of the bands get narrower, and some of the bands get thicker. Now, how does this actually happen? It happens because, well, energy is expended. It does require ATP, and the binding site on actin filaments, there are these binding sites. If they are uncovered, then myosin will attach. If it attaches, it will ratchet, okay, and pull. So it's just going, all these little heads, this is what they do. Every little myosin head, some are pulling this way, some are pulling that way. They're just doing that. Now the interesting thing is that it's already in the locked and ready, and it's it spent the energy to cock it like this. Okay, it spent energy to get ready. It does not spend energy to do that. That's just the release of the energy that was put in. So it puts in potential energy, and then if it can bind, if the binding site gets uncovered, away it goes. So, and then, so it grabs on and does that, but now it's stuck and it needs more ATP to release and reset. So, does that make sense? I've used ATP, I'm ready. The binding site gets uncovered, so I bind, ratchet, and I'm waiting for more ATP. I need more ATP to let go and reset and grab, and if another binding site's available, I'll just grab and do this again. And I'll just keep doing this. But the point that is not intuitive is that the power stroke doesn't use ATP. The ATP was used in cocking the head and getting it ready. So here it is, it's already cleaved the ATP. That's why we have adenosine diphosphate and a phosphate. It's already ready because it's already spent the ATP. It binds, it releases that stored up energy, which kicks off the ADP and the phosphate. It will stay in this position until a new ATP molecule binds with it, which will release it and break that ATP molecule to cock that into place. So, bind and ratchet, get a new ATP to release, and spend the ATP to get into the ready position. If it binds again, it will ratchet and release the ADP, and there it is. This is kind of the final state, and this is contracted, right? It pulled already, and it needs a new ATP to let go and start over. All right, here's more of a three-dimensional view of myosin. So you can see it's technically kind of multiple sorts of tiny fibers together, but that's okay with its little globular heads. And a cross bridge is the connection when the tropomyosin has been pulled out of the way and the binding sites are revealed. The cross bridge is the connection between the myosin head and the actin filament. And here we see the action that this side is pulling zoop, this way, right? Zoop, zoop, and this side's pulling this way. They're both pulling towards the center. And here are the thin myofilaments, the actin. Here's the thick, the myosin. And here they overlapped more. Here is the actual arrangement that I was trying to describe earlier. A thick filament binding to 6 thin filaments, and each thin filament being surrounded by 3 thick filaments. So there are binding sites all around the actin, and there are heads all around the myosin. So there's some redundancy. And there's a cool electron microscope, electron micrograph of a cross section that I'm not going to spend the time to interpret, but through that, looking at that closely, this is what we figured out. That's the bottom line. All right, death. When you die, you will go into a state called rigor mortis, the stiff death, stiffness of death. And it can be very stiff. Now, it will wear off eventually, but this man has been dead for 18 hours and he's very stiff. How is it that he is very stiff? His muscles are in a state of contraction. How could he be contracting his muscles if he's good and dead? I mean, he's obviously very dead here. And I can't remember what I was going to say about football here. So, there's our adenosine triphosphate. Cleaves to his adenosine diphosphate and phosphate molecule. We get that, right? That's the energy currency for the whole body, for every cell, for every cellular action. We're not going to go into how we make ATP just right now, but we make ATP and then we use that ATP in the process of muscle contraction. But how is it used? To release and cock. To release and put potential energy in. The energy that was actually doing the contracting, that was the energy that had been stored there, and then it was ready to go. So the question becomes, what if you run out of ATP? Oh, and this is getting ATP through anaerobic means, pyruvic acid, getting converted to lactic acid, and we're not going to go into that right now, but basically this guy is suffering from lactic acid buildup potentially, because he doesn't have quite the endurance that he needs, perhaps. Well, he went a lot further than I would have gone. So you can go for a long time, right, at this, you can maintain a steady state of high oxygen use, but you actually have an oxygen debt that will get paid back during recovery. So basically, after you run, then you stop and you're breathing until you pay back that oxygen debt and get rid of that lactic acid. Because this can go the other way. Okay, I'm not going to go into right now the high energy intense use of muscles versus long-term aerobic use. We talked about that a little bit on the phone, but I guess I don't have more pictures of dead people in Rigor Mortis, so I'm just going to explain. Let me jump back to dead guy and Rigor Mortis. Maybe that's not a nice picture to stay on. Let me jump back to this one. Okay, so when you die, you stop breathing. Side effect. Your heart stops beating. Another side effect of death. You are no longer delivering oxygen to your cells. Your cells rely on oxygen mostly for making ATP. They can produce ATP anaerobically as well, as long as they've got stored sugar there. You will not be immediately stiff after death because you still have ATP available. And why does that matter? Because you need the ATP to relax. We haven't gone into the calcium, and that's important. The calcium is stored in the sarcoplasmic reticulum. It's released in response to the nervous signal, and it's the calcium that leads to the uncovering of the binding sites and the cross bridges that form. So when you're dead, the calcium can no longer be held in when you run out of ATP, because your sarcoplasmic reticulum was holding calcium against its concentration gradient. It was active transport to hold in large quantities of calcium. When its calcium pumps stop working, calcium's going to start leaking out. When it does so, there's nothing pulling it back in. So it just leaks out. What's it going to do? It's going to bind to the troponin, which makes the tropomyosin slide out of the way and uncover the binding sites. What happens when the binding sites are uncovered? Those myosin heads are going to grab on. What happens when they grab on? They're going to ratchet because they've got energy ready. All they have to do is grab on and then they'll ratchet. Well, they'll keep on ratcheting as long as there's ATP present. But what happens when they're out of ATP? They don't let go, because to let go, you have to have ATP to let go and cock. And so the last thing you do, the last thing your muscles do when they've run out of ATP is one final grab on and do a power stroke. And so all those muscles, all of your muscles, are in a state of contraction. And then they're just stuck there. And eventually your cells just kind of, start falling apart, and the enzymes start breaking everything down, and then those cross bridges will get broken and you will not be stiff anymore. But all right, enough about dying and getting rigor mortis. Let's move on. So this is a look at the axon bulb of a motor neuron. So here's the axon, and it comes down to the axon bulb. And so when an action potential, because you decided to move a muscle, and so here comes the signal very fast, and when it gets to the end of the neuron, it causes release of acetylcholine, a neurotransmitter. And this is at a muscle cell. So here the sarcolemma, the plasma membrane, has receptors for acetylcholine, and that's going to set off the chain of events that leads to muscle contraction right there. So here's a neuromuscular junction, the junction between one neuron and one muscle fiber or one muscle cell. Now this motor neuron branches, and look, here it's branched and it's touching a different muscle fiber. So one neuron cell can excite multiple muscle fibers, multiple muscle cells, but it's all or none, right? If it goes to 5 different cells, then when this nerve cell fires, all 5 muscle cells twitch together. Now you have many, many muscle cells within your muscles. But the difference between a little bit of contraction and a lot of contraction is how many different muscle cells are being stimulated at once. Okay, so again, muscle or neuron with action potential causes release of neurotransmitters. Now these neurotransmitters don't stick around for long. They get quickly taken back up. So, your muscles don't get a signal and then stay contracted, because the signal is very, very brief. But what happens in response to the signal? It's going to change the voltage on the cell membrane. It's going to open, they're ligand-gated ion channels, channels that open and allow ions in response to a ligand, in response to some small thing that they bound with. And we haven't talked about nervous system. It's basically the same thing as the nervous system, and we'll go over action potentials there in detail. But it's this depolarization of the membrane that is the signal, the start signal. Now, here it's happening just at this neuromuscular junction. Well, we got to tell the whole cell to contract, and it's a very long cell. So how are we going to spread that signal? Well, here it's traveling, a wave of action potential spreads very quickly along the whole cell. And remember the transverse tubules? These are extensions of the cell membrane. And I might have accidentally said they were extensions of the endoplasmic reticulum. I take that back. They are extensions of the cell membrane, and they're going down into or around the muscle cell, around the muscle fiber. So down they go, carrying the wave of action potential, this depolarization. So basically it's a little switch, okay, a little switch, because it's a change in voltage. What does that do? That stimulates this sarcoplasmic reticulum, which is the fancy word for an endoplasmic reticulum in a muscle cell, and in response to that, it releases calcium ions, which it has kept in storage. It's always actively pumping calcium ions in, but when it gets that little switch, that little change in voltage, it opens up the floodgates and lets calcium flood out. So calcium goes out, binds to the tropomyosin, that little rope that covers the binding sites. It binds to a specific place called troponin. So here comes calcium ions, binds to troponin, which causes, and you can see the troponins covering these yellow little dots, those binding sites. And so it just changes the conformation of the protein, and it just kind of changes the shape of the rope, so that these binding sites are uncovered and cross bridges can form. And it all happens super fast, and the calcium gets taken back up super fast. That's the part that's hard to understand, is step after step after step after step, and then backwards, and it all happened in milliseconds. So it's crazy to think about. But again, here's the tropomyosin, which is this ropey protein covering up the binding sites so that myosin heads cannot bind to them. In the presence of calcium, whoosh, ta-da, I've uncovered, and myosin immediately grabs on. Can't help it, just immediately grabs on. And if it grabs on, it's immediately going to start ratcheting for the whole time that those binding sites are uncovered. But the calcium gets taken up very quickly, so unless the nervous system signal is still there, saying contract, 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 it just very quickly is going to relax. And this happens so fast, and your muscles are twitching all the time, and you've always got some muscles in a state, some muscle cells just twitch, 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 just to help you, stay upright or help your head from falling down. So it is hard to understand because it's all happening so quickly and there's so many little steps to it. But things are just very strange at the molecular level. I don't know how to make them unstrange. So the calcium gets pulled back into the endoplasmic reticulum very quickly, and with it gone, the binding sites get covered back up, and the muscle will relax. Whew, that was a lot. So take it easy. There are toxins that affect muscles, causing different types of paralysis. So curare is, they boil these vines, and anyway, it will suffocate by, well, your respiratory muscles will stop working, and basically, is this curare that, I don't think this is curare, but there are different ways. I mean, basically, here's the acetylcholine that gets released. What if you cover up the receptor? Well, then the muscle won't contract. Tetanus and botulism kind of have opposite effects, right? Tetanus, everything's contracted. Botulism, this is not either one. This is myasthenia gravis, and I think, well, they did a short-acting acetylcholinesterase, something to break down the acetylcholine, and it restored muscle tone. I think that they were in a state of tetany until their muscles just fatigued. But I better not. There's some things I haven't reviewed, okay? And so I'm not going to tell those stories. We're just going to end with the different toxins. Either can cause, the botulinum toxin causes muscle paralysis that's floppy, like the muscles just don't get the signal to contract and they just cannot contract. And the tetanus toxin is just the opposite, the muscles cannot relax, so the acetylcholine just stays bound to the receptors on the muscle cells, so the muscles just fire, and they cannot relax. All right, watch the videos. Hopefully what I've said helps rather than hurts.